Hey everyone, how you guys doing?
Good.
We still have a couple of people missing. Missing a couple of the presenters today. Okay. Can everyone see my screen?
Yeah.
Okay. So typical announcements. So quiz 9, since I went through a lot of material last week, I decided to give everyone who was in our Zoom session the quiz and attendance points completely. So the quiz 9 was counted as from attendance last week. Okay. Quiz 10 will be out tonight. If you haven't done so already, please upload your presentations. I cannot give you a grade without them. If you notice you don't have a grade and you already presented, make sure that you uploaded your presentation. So next week, November 11th is one of those days where I have to go asynchronous with you guys. You can either come to my later sessions or you can just watch the video, do the quiz, okay? We cool?
Yeah.
Okay. So we'll go into presentations now. Let me just allow sharing. Okay. Who wants to go first?
I can go.
Okay. Oh, are we dueling each other or?
I'll go.
Okay.
Can you see my screen?
Yes.
Okay. Okay, so my question was to label the spatial and polarity relationships among the following translational components: the ribosome, the small and large subunits, the growing polypeptide chains, amino acid attachment to tRNA, mRNA codon, and anticodon. So for the background information, the mRNA carries the genetic code to the ribosome from the DNA in the nucleus after transcription. The ribosome has two subunits and is called the small and the large subunit, and it reads the information on the mRNA from the 5' end to the 3' end. During translation, the genetic message on the mRNA is decoded in the small subunit when the mRNA is paired with the anticodon on the correct tRNA through base pairing. The anticodon is the sequence that is complementary to the codon on the mRNA. The tRNA is also bound to the corresponding amino acid using covalent bonding. The base pairing that is happening between the tRNA and the mRNA is holding the amino acids together so that a peptide bond can form. The formation of the peptide bond is taking place on the active site in the large subunit of the ribosome. And then this is the answer to the question that I had. This right here is the large subunit of the ribosome, and this is the small subunit of the ribosome. Right here, you see the mRNA with the codon, and here you see the tRNA bringing the anticodon to bind to the codon on the mRNA, and the amino acid here that the tRNA also brought along with it. And then here is the growing polypeptide chain.
Good.
That's it.
Everyone cool with that figure?
Yeah.
Okay. Okay.
Okay, so I had a question to briefly explain what occurs at each of the following ribosomal sites, the A site and the P site. So some background information. Translation of the mRNA can be divided into 3 steps: initiation, elongation, and termination. However, the A and P sites are more used in the elongation step of translation, where the polypeptide chain is produced. Out of the 3 subunits in the ribosome, the aminoacyl A site is first, the peptidyl P site is second, and the exit E site is last. So the A and P site, it gave me a lot of information, so I tried to summarize it as much as I could. In the A site, it's the arrival site of each charged tRNA that is attached to an amino acid later to be added to the polypeptide chain. In the P site, it releases energy, binds to the tRNA, which forms the polypeptide bond, and it holds that growing chain. After in the termination step, the release factor RF3 binds to the ribosome. tRNA is released from the P site and let out to the other subunits. And that's all.
Good. Does that make sense to everyone?
Yeah.
I think we're missing 2 people who were supposed to present. So, okay, we'll continue with my presentation. So, if they join in later, they can present them. If not, we'll see what happens. Okay. So, let me just get this done. Okay. So today we're just going to cover a bunch of questions that are related to transcription and translation. Okay, so for one thing, where are you guys in lecture for translation? I know you guys started it, but where are you guys now
We just started translation
today? Okay, good. At least with the questions I'm going to be covering, it's pretty much what I covered last week too, so we should be okay, but we'll cover more stuff if we need it. So this question is more in transcription side. So in the following coding strand of DNA, exons are colored in black and introns are colored in red. So this is the sequence. We have exon, exon, intron. What is the sequence of the complementary pre-mRNA? So what do you guys think? Any ideas? No? Okay. So let me walk you through this one. So when it's asking for the sequence, we look, so what do we know? We know that it is the coding strand. So there are differences when it comes to being the coding strand and a template strand. So in this case, we'll see what goes on with the coding strand. So to get the pre-mRNA of the coding strand, you just have to change the thymines to uracils, okay? So in this case, we have this coding strand DNA sequence, right? We just copy and paste that guy. We're like, okay, we have that. When we transcribe it, we get this, right? So in this case, since it's the coding sequence, you're just changing the T's to U's, right? So that's done, right? Does that make sense?
Yes.
Okay. Next, this is another question where hold on. So considering the following sequence of DNA, assume that the top strand is the template strand used by RNA polymerase. Draw the RNA transcribed, label its 5' and 3' ends. So what do we know? We know that the top strand here is the template strand. So when a template strand is involved, you use the complementary strand to transcribe, to get the transcribed mRNA. Okay, so if this complementary sequence wasn't given and this was the template strand of the DNA for RNA transcription, you would find the complementary strand and then you would go into transcribing that. So this ends up the coding strand, and then you change the T's to a U, and that's how you get the pre-mRNA sequence. Are we okay with this?
Yeah.
Hey, can you go over that one more time, please? That last part.
This part here?
Yeah, the second.
Here? Okay.
Yeah.
So when you have a question saying the template strand, that this is a template strand, and you're like, okay, so with DNA, one strand is the coding strand and the other strand is the template strand. So if it says like coding strand, we have the coding strand, right? So when you do the coding strand, you just change the thymines to uracils. Whereas with the template strands, if you're only given just a template strand, you get the you take the complementary to so complementary as in the other strand of that DNA molecule, and then you would transcribe that complementary strand. So that would be your coding strand, and then you would take that and then you would put U's instead of T's for the mRNA, pre-mRNA. Does that make sense?
How'd you get that the one at the bottom?
So it's just replacing this guy, right?
Yeah, and then you flipped it around?
Yeah.
Okay.
Yep. Because you always read DNA or RNA from 5' to 3', right? So in this case, remember the DNA sequences and their strands are anti-parallel, right? So you would never put a combination of those guys 5', 5'. You would put a 5', 3', right?
Yeah.
So when you do that, you have to flip it around, because when you're doing when you're writing out DNA, if 5' and 3' wasn't written out, it would be automatically assumed that it's read in 5' to 3'. In this case, we specifically say this is our 3' end, right? So that's only reason why we're reading this as 3' to 5'. If this didn't exist, like these letters or numbers didn't exist, we would be reading this as 5' to 3'. Are we cool with this?
Okay.
Okay, so suppose that a portion of double-stranded DNA in the middle of a large gene is transcribed by an RNA polymerase. As the polymerase moves through the sequence of 6 bases shown in the diagram below, what is the corresponding sequence of bases in the RNA that is produced? So what do we know? We have both given, right? We have a coding strand and a template strand. Like, okay. So what do we use to produce our mRNA?
Use the template.
You would go with the coding strand because we already have the coding strand given, right? Does that make sense why we chose the coding strand? Because it's already given to us, right? The template strand, when it's only the template strand given, that's when you find the complementary, which is the coding strand. And then you would go into transcription. So in this case, we choose the coding strand. You end up with this answer, guys. So you have U, G, U, G, A, G, C, C, right? Because you're only replacing the thymine here. So that's why the answer is that. And this is in 5' to 3' direction. Does that make sense?
Yeah.
Okay. So again, make sure to have it written 5' to 3' if you haven't labeled them that way, or you'll get it wrong, which can be a pain in the neck sometimes, but okay. The genetic code is blank, meaning that the amino acid may be encoded by more than one codon. So what do you guys think?
Universal?
Okay. So let's go over some definitions, I guess. So with the features of genetic code, we have, so the first one we have is degenerate. Degenerate means that more than one triplet codon can specify for one amino acid. And then you have the second one, which is unambiguous. So each triplet can only specify one amino acid, right? Then you have commaless. So commaless just means that once it reads, once it starts beginning translation, the codons are read sequentially without any breaks, unless you hit a stop codon. But even then, if a stop codon is in the middle of a sequence of 4 codons, multiple codons, they're around them. If it hits a stop codon, it stops right there, doesn't go anywhere, and it does not restart, okay? And then you have non-overlapping. So any single ribonucleotide within the mRNA is part of only one triplet. So it's not going to be like ACA, ACA, UGA, those are two different codons, and they don't overlap with each other, right? Does that make sense? And then with the genetic code, it's not completely universal, but it's nearly universal. So in this case, a single coding dictionary is actually used by viruses, prokaryotes, and archaea, and eukaryotes. There are some differences, but it's pretty much universal, but not completely universal.
What is a single coding dictionary?
Essentially, okay, so what that just means is like they use the same codons to translate into your amino acids. Does that make sense?
One codon for one amino acid?
So in this case, it's just saying that, you know how we have the genetic code table, right?
Yeah.
That genetic code table is pretty much like the single coding dictionary. And that is kind of like kept between each of these guys, the viruses, the prokaryotes, archaea, and eukaryotes. Does that make sense?
Wait, I don't understand. There's one column for the amino acids.
Okay, so you know how we have the genetic code table, right? You can
Yeah.
use that between all of these different species or kingdoms, I guess. Does that make sense?
Okay. So wait, the viruses, prokaryotes, and archaea, eukaryotes always use the same coding dictionary?
Essentially, yes. That
Uh.
genetic code table. Okay. So that makes the answer to this degenerate, because degeneracy is the term for redundancy. Or in this case, an amino acid can be encoded by more than one codon. Does this make sense? Yes, no, maybe so.
Yeah.
So the sigma subunit of the bacterial RNA polymerase does what? What happens? What does it help the polymerase do? Anybody have any ideas?
Is it A?
Yeah, it is A. So the sigma itself is a polypeptide unit of the RNA polymerase that binds to the region on the DNA, so the promoter. So this area is actually the promoter area, and it allows, it puts a catalytic center of the enzyme in contact with the initiation site of that gene. Does that make sense?
Yes.
In this interpretive drawing of the heteroduplex formed between mRNA, so this here is mRNA in orange, and it's coding DNA in pink. What do the loops labeled A through G represent? what do you guys think? So we have all these loops going on, right? So all these loops, we have something. So we have a DNA strand and an mRNA strand. So mRNA, in this case, fully spliced, right? So it's trying to bond with each other. So what are these loops? A, B, C, D, E, and F, and G. What do you guys think those are? Okay, what do mature mRNAs don't have? Any ideas? What are we splicing out from pre-mRNA to mRNA?
Is it introns?
Yep. So essentially in mature mRNAs, so in this case, it is a mature mRNA, and the introns have already been spliced out and the exons are fused together. So when they try to bind, the exons can find their matches, but the introns present in that DNA can't. So they end up looping out. So that's why those guys are introns, right? Does that make sense?
I'm kind of confused.
Okay, where am I losing you?
Because it's like, if the introns were spliced out already,
Mm-hmm.
then how is it looping? Like, how is it causing it to form the loop? Like, is it, like, I don't get that.
So in this case, they're saying that it's a heteroduplex formed between DNA and mRNA. So the DNA still has the introns still in there, the sequence for the introns. The exons are binding to their complementary sequence on the DNA. But then the introns are like, it doesn't exist. So they're like looping out, like trying to push it away that the exons can fit together properly. Does that make sense?
Yeah, I get it now. Thank you.
Okay, good. Any other questions? Okay. The poly A tail of mRNAs do what? What do you guys think? I don't remember how much we covered with this guy, or at least how much Dr. Osuna covered. But what do you guys think? Okay, I'm gonna hear crickets if we get any quieter. Okay, so with poly A tails, as we know, so poly A tails, they are added to the 3' end on most eukaryotic mRNAs, and it's typically after transcription that they add it. And this is to actually prevent the degradation of the mRNAs. So the answer is all of the above, right? Does that make sense? So it's essentially stabilizing these mRNAs against degradation by exonucleases. Or they'll just chomp it all up. Does that make sense though? What the poly A tail is about?
Yeah.
Okay, so what is true about tRNAs in this case? What do you guys remember? I know it was a lot of information last week, but no,
Is it 4?
they can only carry one amino acid per molecule, not
Okay.
more. Okay. So
Is
essentially,
it one?
yes, it is one. So tRNAs do have double-stranded regions. So these guys like this. So even though it's not two different strands, they come together and form that double-stranded region. So they end up folding back, they fold back and base pair with the other regions of the same molecules, creating the secondary strand, the four-leaf clover shape, right? Does that make sense? Yes, no, maybe?
Yeah.
An amino acyl tRNA synthase for valine is mutated so that it now attaches the amino acid glycine to the tRNA valine. Instead of valine, what will happen at translation? What do you guys think? So choice 1, there will be glycines at all valine positions and valines at all glycine positions. B is saying there will be valines at all glycine positions and glycines at all valine positions. Then you have choice C, which is there will be valines at all glycine positions and at all valine positions. Then there will be glycines at all glycine positions and at all valine positions. And E, there will be no translation. So what do you guys think will happen? So the first sentence is kind of telling you part of what is this mutated tRNA synthase is up to. So what happens? Guys, any ideas?
Is it D?
Yes. So it is D because the tRNA valine will still recognize that codon, but the codon, it's the tRNA will have glycine attached to that codon instead of valine. Does that make sense? And any
How-
glycine codons will end up just being recognized by the tRNA glycine. Okay?
How do you figure this out? Do you have to look at the chart?
So in this case, you don't even have to look at the chart just because the question is saying an amino acid tRNA synthase. So essentially it's the tRNA that is charged with valine, right? That guy is mutated so that it now attaches the amino acid glycine instead of valine, right? So what does tRNA synthase valine do? It attaches valine, but now it's mutated. I was like, okay, it's adding glycine instead of valine, right? So in this case, the only thing you need to know is that, okay, so instead of valine, glycine is getting added to all valine positions. And then at the end of the day, glycine positions, if there's a codon for glycine going on, it will be recognized by the tRNA synthase for glycine. Does that make sense?
Yeah.
Okay, good, good, good. So that's why the answer is D. Okay. Okay, so during bacterial translation initiation, the mRNA blank, what does it do? So what happens when they first start off with mRNA? Or what happens in bacterial translation initiation? Any ideas? Which word out of these guys is screaming bacterial translation or prokaryotic translation initiation? There are a couple of words that are screaming prokaryotic translation initiation. Do you guys remember what happens in prokaryotic initiation translation? Yes.
Is it the D? The
Yes.
Shine
Yep. So when you see Shine-Dalgarno sequence, that's prokaryotic or bacterial translation initiation, okay? So essentially, translation initiation involves the binding of mRNA to the small subunit through the base pairing of its Shine-Dalgarno sequence to the 16S ribosomal RNA of the small subunit, okay? And this allows that to be oriented towards the start codon or the AUG codon in the P site. Then this FMET tRNA comes in, binds to the AUG codon, and then the large subunit comes and attaches. This part requires some hydrolysis of GTP, but that's how that gets started. So that is why the answer is D. So the first thing that needs to happen is that the mRNA sequence has to bind to that sequence and orient itself towards the start codon. Does that make sense?
Yeah.
Okay. During elongation phase of translation, charged TNA enter the ribosome at the blank.
It's a C.
Yes. So again, the A site or the amino acid site, not the amino acid site, is so that's where the charged tRNAs come in. They enter the ribosomes and then deliver that correct amino acid. The P site is where the growing peptide bond goes on. And then the E site allows that uncharged tRNA to go out of the ribosome and then get replenished, right? So that's why the correct answer is A site. Okay, so I don't think I specifically said peptidyl transferase, but what do you guys think that does? Okay, this might not be the easier ones. So at first, peptidyl transferase was thought to be a protein enzyme, but in this case, it's actually a ribozyme or an RNA molecule that has catalytic activity. In this case, the bacterial ribosomes, they assign this function to this specific ribosomal RNA, and it allows, it catalyzes the formation of peptide bonds. So whenever you think peptidyl transferase, think peptide bonds because it's saying peptidyl transferase. So it's allowing the formation of peptide bonds between the different amino acids during elongation. So it's essentially, this name is actually a function of a ribosomal RNA in the large ribosomal subunit. Does that make sense?
Yes.
Okay, good. In this drawing of hemoglobin, what levels of structures are visible? What do you guys think?
Tertiary. Tertiary. C.
Okay, so with this, so in general, when you look at these kind of proteins, you actually see secondary, tertiary, and quaternary. Because hemoglobin itself is its own, so we have the heme, we have the other components. So like these folds over here can be considered as secondary. We have the tertiaries going on with like the alpha chains and the beta chains and all that fun stuff. And then you see multiple different subunits interacting with each other to form that functional molecule. Does that make sense? And essentially, when you look at a protein itself, you won't be able to see the sequence of amino acids right away. Does that make sense?
Yeah.
Okay. If there were 6 bases in DNA and still only 20 amino acids, how long would a codon need to be to specify an amino acid? Do you guys have any ideas? I don't think he went over this. Okay, so to do this, it's essentially, so essentially to figure out the number of codons that we can get is the number of bases to the power of the length of the codon. So in this scenario, so when we look at our genetic code table, we have about 64 combinations of codons, right? That's because you have 4 bases and you take that to, so each of them can kind of go up to like, so in this case, we have the length of the codon as 3. That's how you get 64, because you take 4 to the 3rd power. That's how you get 64. So in this case here, in this question here, you have 6 bases in a 2-base codon. So in this case, we're kind of testing out which ones work. So if you have 6 bases with a 2-base codon, you get 6 squared, which is 36 combinations. So essentially, this would be a minimum of number of combinations that you need to form 20 amino acids. So in this case, since you have 36 different combinations of codons, that means you can have enough to encode all the 20 amino acids that we need and the stop codons. Does that make sense? And that's why it's 2.
I don't understand how you got that. Can you go over that again?
So with this, we're looking at, so this question, if there were 6 bases in DNA and still only 20 amino acids, how long would a codon need to be in order to specify an amino acid? So in this case, the way we would look at the, so we need to figure out.
Is it the length of the codon that you're looking at in the equation?
Mm-hmm. So essentially, so with this kind of question, since you don't have the number of combinations given, it's essentially plugging that number in and seeing which gives you higher than minimum 20 amino acids and at least 1 stop codon, right? So you need at least above 21 codons, right? So in this case, the first time you try with 2, you already get 36 different combinations. That's what's giving you, that's how you end up with 2 bases. Because when you already have 2 bases in the length of the codon, that's what, that gives you more than enough codons to actually encode for all the 20 amino acids and the stop codons. Does that make sense?
Okay.
Okay. A nonsense suppressor mutation is a mutation that does what? Okay. So essentially with this guy, a nonsense mutation ends up changing an amino acid that codes, so a codon that codes for an amino acid into a termination or a stop codon, right? So in this case, the second mutation ends up suppressing that. So in this case, so we understand what a nonsense mutation is. Now we have another mutation that is suppressing the effect of this one, right? So essentially, the suppressor mutation would end up restoring the functionality of the nonsense mutation by allowing an amino acid to be added to the polypeptide. So in this case, it is a mutation. Yeah, it's essentially recognizing that codon instead. Okay. I need to look at the wording of this question again. Okay, so I think the wording of this is wrong, but essentially what a nonsense repressor mutation does is that it restores that amino acid that they're coding for. Does that make sense? Yes, no, maybe so.
I think so.
So do we have our remaining? Okay. Do you guys have any questions? Concerns?
Are we gonna do something about the exam? Like go over a little bit? Or did we already do that?
No. But if you guys have any questions, I guess send me an email of the questions by like tonight or so, and I'll put up a video on it because I don't want to dwell on the exam too much at this point.
Okay.
I'll put up a video on the questions that you guys have questions on, but you guys gotta send me which questions you guys want me to put in.
Yeah, I guess part of it was that we were not all the way sure what would be like on the exam, so it'd be nice if we knew what was most important
Oh,
to,
you mean like for the next exam?
well, yeah, this one coming up, isn't that next Wednesday?
I honestly gotta have no idea.
Okay.
So all over the place. But let me just double check that. Give me 1 second.
It's next Wednesday.
It is?
Yes.
Oh,
He sent out an email.
you did? How did I miss that? Oh, okay. Chapter 17. Ooh, okay, we're not meeting before that. Okay. Well, that would have been good to know a little ahead of time instead of right now. Okay. Definitely go to the review session if you can with Dr. Osuna, because the exam's gonna happen before we meet again, and we won't be able to meet afterwards because my meeting being annoying and annoying times. So
Okay, what about some of us who can't make it to the review session? It might be recorded, but it might not be released or until, too late or.
okay. I'll try to get some stuff up for chapter 17, maybe by the weekend.
Okay.
That way you guys have something to look at. I'd have to check which is actually chapter 17. Hold on. Forgive me. It's been a little bit all over the place lately. Okay. Okay, I'll have a video on. Okay, so chapter 17 is regulation of gene expression in eukaryotes. So I'll have a video or a study guide up. It might be more detailed than what he's asking for, but make sure you guys look at his lecture too, and then study that way, okay?
Okay.
Are we all okay? Are you guys freaking out?
A little.
Don't be freaked out. I know it's easier said than done, but you guys got this. Okay?